Bismillah ar-Rahman Rahim. We start the new uploads for this coming CIE exams, and these are the 2022 uploads. And this will, of course, be on the new syllabus. So I'm very excited to be uploading all these new videos, and I wish you all the very best in your preparation and in the exams. I will be starting from the last CIE exam, which was the November 21 exam. So we will start with that. And this paper is 1 hour 15 minutes. This is the paper 2. Which I will three variants, and I'll be discussing all the three variants so that you all get a very good practice in all these questions and inshallah do very well. Uh, as we go on to this paper, the first question, as you can see, is question 1A, figure 1.1 is a transmission electron micrograph of cells from the leaf of a plant. Now remember, transmission electron micrograph, and the other one is called the scanning electron micrograph. Now scanning means it just scans the surface, so you only see the surface features. But now you're looking inside it, so you can see the details of it inside. And then of course it says name the cell structures. Name the cell structures X, Y, and Z. So X, as you can see, this is a very large nucleus here, which is a very, very typical mottled appearance. And you can see this is the nucleus. And then the Y is the chloroplast. And Z is the vacuole. Because why did we not say cell sap? Because that actually is the cell sap, but it says name the cell structures. We didn't say name the contents. We said name the cell structures. Now, at the end of this paper, I'll go through some micrographs just to quickly revise this micrograph story because you all are a little not clear on that. I can see it. So then coming on to X, Y, and Z, now it says state two ways in which the structure of an animal cell. State two ways in which the structure of an animal cell differs from plant cells. Structure. Now, which one has been mentioned before? Animal cell is mentioned before. So this is the first one, and then this is the second one which is being compared. So if I say you compare Ibrahim with Fahim, then you talk about Ibrahim, and that's what we are going to expect you to talk about. So I want you to say this: what is in an animal cell? So when you look at the animal cell now, as we can see very clearly, that there is no large permanent vacuole. I'm not writing on the lines because I have a lot to write. No large permanent vacuole in the animal cell, right? Then there are no chloroplasts. No chloroplast in the animal cell, right? No cell wall in the animal cell. That was a very basic one. I don't think anybody could have lost marks on this because I'm sure you all are pretty brilliant and you know all these answers. So no plasmodesmata. Yeah, you know that there's no plasmodesmata in it. So then you have in the animal cell, you have glycogen granules. So animal cell will have glycogen granules, while the plant cell will have starch granules. Right? Now, another thing which you could have said is that the nucleus in an animal cell is central. Now, what does that mean? It means that if this is an animal cell, the nucleus is in the center somewhere here. This is the nucleus. While in a plant cell, it is because of the vacuole, the nucleus is pushed to the side. So in an animal cell, the nucleus is in the center. This is an animal cell. So the nucleus is central, or you could have said nucleus is not at the edge of the cell or at the periphery of the cell. Periphery, shayad afridi caught a catch at the periphery. So, and then the last point is you could have said is centrioles are present. Centrioles present. So this is all what you could have said, and that was the correct answer. Then coming to question number B, one cell structure Y in figure 1.1 contains a large starch granule. Yeah, starch granules are found in the chloroplast. So name the chemical reagent used to test for starch and state the color change. Now you did not read that, and you're going to make a big mess of this if you don't do it correctly. The reagent, of course, is iodine solution. There's no doubt about that. You would know this, but if you just wrote iodine, I'm sorry, you won't get your mark for that. It is an iodine solution in potassium. It is iodine in potassium iodide. So you could have said that also. Now here is the thing where you would lose the mark, because you would all say blue black. Now it's not actually correct. It says color change. What was the color before and what was the color after? So what was the color before and what was the color after? And so the change in color would be orange, or if you said brown, to the color changes from orange or brown, it changes to blue or black. Or you could have said blue, black, or purple even was correct. There's a slash everywhere. Blue, black, purple. But you see, where you would have gone wrong is if you said the color would change to black. Now that would be, there we would cost a mark for that. You won't get a mark for that. So orange, brown to blue, black, or purple would get you the mark for that. Then it says starch granules contain amylose and amylopectin. Right? Now describe the similarities and differences. Similarities and differences between the structure of amylose and the structure of amylopectin. Now, unless you know this is something which is very basic. You've got to really know it, and then only you could have done it. Now, you can't be really coming up with just saying, okay, well, I didn't revise this and all that. No, no, I'm sorry. This is not a paper four. Paper four is a lot of in which you get questions in which are very, not very much from the book, but actually just test a lot of your biological knowledge. So coming to the differences, I'm just going to give you all the differences. So similarities are, let's look at the similarities first. So I usually give you a heading and I say, oh, do that in on the paper as well. Just write similarities. So both are polymers or both are polysaccharides. Both are polysaccharides. That got you a mark. Then both have alpha glucose. Both have alpha glucose. I'm sure you all remember that. Both all are joined by glycosidic bonds. So they have glycosidic bonds in both of them. Both have glycosidic bonds. I'm sure everybody knows that. And then they all contain 1,4 glycosidic bond. 1,4 alpha glycosidic or glycosidic would be fine. So these are the similarities. Now you can see how I have grouped them, and of course I try to group it for you also. Similarities are that they are both polysaccharides, alpha glucose joined by glycosidic bonds, 1 to 1,4 glycosidic bonds present in both. Differences: amylose molecules are unbranched, amylopectin has 1,6, amylose is helical, amylose is a smaller molecule, and amylose is more compact. What does the word compact means? Compact means it's in a small space, it occupies very little space. Like we say, oh my God, this house is very compact, so you got everything very much at hand. So the word compact is a word which you must understand whenever we use that word. That means it's sort of everything is stored in a very small, in a very neat and tidy manner. So compact is a word which will be often used here in biology. So just go through this. If there is any point that you do not know, then you need to look up the structure of it and be clear by what do I mean. Amylose is helical. Helical means it's like this, helical. Then amylose is a smaller molecule as compared to amylopectin. Why? Because amylopectin is a lot of branching. So it is a much larger molecule. So this is what you need to revise and then go through if you're not very clear on anything. Then coming to question number 2a, figure 2.1 is a transmission electron micrograph of a transfer section through a blood capillary that is surrounded by actively respiring cells. So more respiration means more carbon dioxide being produced, more oxygen being taken up. And then it says identify cell Q shown in figure 2.1. Now if it's a capillary, well it has to be, you see, now look here, now this is the thing which you need to notice. The capillary, this is the wall of the capillary. So what is this? There's a nucleus, and what is this nucleus in? This is a nucleus, must be an endothelial cell. So there's a nucleus of the endothelium. And I know I gave a paper the other day in my school, and many of the students, instead of endothelium, wrote epithelium. And I said to them, and I joked with them, and I said, you know, the endothelium is very angry because you called it epithelium. It's just like calling Ibrahim Fahim. So endothelium, you've got to be realizing that the cells, the wall of the capillary wall is one cell thick, and it's an endothelium. The cells of the endothelium. So identify cell Q shown in figure 2.1, and there's a red blood cell. Then it says fluid R is formed from blood, from the capillary. So fluid R is from the blood. As soon as the blood is inside the blood, we would say that would be the plasma. And as soon as that fluid comes out, it is called tissue fluid. Of course, you can't use the word TF, I'm just saying that it's a tissue fluid. So fluid R is formed from blood in the blood capillary. State and explain one difference between the composition of fluid R and the composition of the blood. So basically, what are they asking you? They're asking you the difference between plasma and tissue fluid. Because as soon as that liquid was inside the capillary, it was called the plasma. And as soon as it's come out, that same fluid is called tissue fluid. So state and explain one difference between the composition of fluid R and the composition of the blood in the capillary. Now you can see how what you could have written. The difference was you could have said no plasma proteins. So there is no plasma proteins in the tissue fluid as compared to the plasma. And plasma proteins, there are lots of plasma proteins. But why? Why are they not there? Number one, they're large molecules, so they cannot be filtered through the gaps between the endothelial cells. So there are no plasma proteins, no red blood cell, no white blood cell, no platelets. And the reason being is that, they cannot pass through the gaps between the endothelial cells in the wall of the capillary, or you could have said in the endothelial wall. So this is what you have to say for a difference in explanation. But of course, there are many differences in explanation which are given to you in the mark scheme, and I'll just discuss one or two more. Another possibility is you could have said that the tissue fluid has a higher partial pressure of carbon dioxide. Now you've got to understand what I'm talking about. What I'm talking about is that this is the cell, which is an actively respiring cell. So a lot of carbon dioxide is produced, so it goes into the tissue fluid. But before it can go into the capillary, this is the capillary which I'm showing with the dotted line. So tissue fluid is this fluid outside the cell. This is the tissue fluid outside the cell. This is the tissue fluid. So there's more carbon dioxide. So tissue fluid has a higher partial pressure of carbon dioxide. Why? Because it is produced by the respiring cells. Of course, as soon as the carbon dioxide will diffuse into this, then of course, but it will take some time for it to diffuse. I'm not saying a lot of time. So this is produced by the respiring cells. Another comparison which you could have done is that the tissue fluid has lower partial pressure of oxygen. Naturally, what you've got to understand is that this is the capillary. And inside the capillary, you have the red blood cell, and the red blood cells contains the oxyhemoglobin. And this is the cell outside. And so oxygen is going to be higher there, and then oxygen is going to diffuse, and this is the tissue fluid. And then it's going to move into the cell. It's going to diffuse into the cell. Don't say the word move. Move should not be used at all in biology. We should just say diffused. So tissue fluid has lower partial pressure of oxygen. So oxygenated blood from the lungs is coming, and the blood, of course, carries the oxygenated blood from the lungs. In the red blood cell, we've got the oxyhemoglobin. So please understand this point as well, which you could have also given. There are a number of points which you could have given, and there are many alternative mark schemes for this. The last point which I want you to remember is that tissue fluid has a higher concentration of glucose. Now, the higher concentration of glucose, why would the tissue fluid have a higher concentration of glucose? Why is it a higher concentration? But it is carried by the blood to the respiring cells. So as soon as the blood is going to diffuse out and enter the tissue fluid, and then of course it's going to be used by the cell. So carried by blood to the respiring cells, it is small, it can be filtered through the gaps, and then it is filtered out under pressure from the plasma. So the pressure is going to cause the glucose to diffuse out. So it's going to diffuse out through these gaps. And the gaps are the very important thing which you've got to understand. This diffusion, you can also say diffusion through the cells in the capillary wall. Then coming to question B, part 1, the cell surface membrane of the respiring cells have a role in regulating the exchange of substances between the cells of the body and the blood. Draw a diagram in the space below to show the arrangement of the phospholipid molecules in the cell membrane. Always look at the marks, 2 marks. So you've got to be very clear in drawing the phosphate head and then the fatty acid tails, and then pointing inwards, the tails are towards the inside. So this is what we had to say. We just have to draw these. Just draw these. Simple. So the phosphate heads would be on the outside. So bilayer drawn with heads pointed outwards and the tail pointing inwards. Phospholipid drawn with one phosphate head and two fatty acid tails. So you drew the two fatty acid tails. These are the sort of the representative diagram which you drew. So this was, now I know of a student, I remember checking a paper and he had done this. So I mean, of course he was chungar, but I mean, he knew there was a phosphate head, but he thought there was just one one tail to it. So, I mean, this is of course all a lot of beautiful story, BS, but that would be wrong. So 2 marks for you to getting this, showing a bilayer and showing one phosphate head and two fatty acid tails, right? Part 2, cholesterol is a molecule found in the cell surface membrane. Describe one role of cholesterol in the cell's surface membrane. That's a 1 marks. And then this is, please remember, this has been out of the syllabus, so you do not talk about nicotine anymore. Nicotine has been removed from the syllabus, no longer in the syllabus, in 2022 syllabus. Now, the role of cholesterol, basically what you've got to understand is maintain, or you use the word regulates fluidity. Remember this word, just remember it, fluidity, and maintains and regulates stability. So two words to remember, fluidity and stability. And you can say regulate or you can say maintain. Whatever comes to your mind, remember one. I don't want you to remembering both of them. So maintains or regulates stability and fluidity was the only mark scheme points. As we look at question number 3, cholera is an infectious disease caused by the bacterial pathogen Vibrio cholera. So you have to know these names. Please remember the spelling of cholera is AE at the end. Describe, sometimes they've asked you for the name as well, so you must remember that. Describe how Vibrio cholera is transmitted from an infected person to an uninfected person. Basically, you know that the patient who has cholera, or the person who has cholera, the bacteria would be passed out in his feces, and the feces must contaminate a water source or must contaminate the food. I mean, how is that possible? Yes, it is possible if the feces are washed into rivers and lakes and people drink water from those rivers and lakes. Number one. Number two, if the flies sit on the feces and then sit on your food and then you eat that food, well, that's how you're going to get cholera. And this usually happens after natural disasters, because that is when, all the sewage lines and the water lines are all, broken, and there's a lot of mixing of it. And that is when people get a lot of cholera after natural disasters. So basically, you have to understand is how it is transmitted. As you can see, the wordings are very clear. Feces contaminate drinking water, or you say eating food washed in contaminated water. Like, for instance, if you wash salad, now you don't cook the salad. So if you washed lettuce, the salad lettuce leaves, and you wash it with contaminated water, you don't cook lettuce. So if you're eating that, well, you would get the disease, because the contaminated water would contain the bacteria, which is Vibrio cholera, which causes cholera. Vibrio cholera releases a toxin called collagen. Collagen is a protein molecule made up of 6 polypeptides. So it's got a quaternary structure. Figure 3.1 is a ribbon diagram of a molecule of collagen. Look at the diagram and you can see the T is the alpha helix, which is of course the secondary structure which they are trying to ask you to understand. Then they've asked you to describe what is shown by the part of the molecule labeled T. Well, what have they shown you by the part? It's an alpha helix. And the alpha helix is the secondary structure. Alpha helix is the secondary structure. So look at the marks, 3 marks. Then the hydrogen bonding. The hydrogen bonding. And that is the one which causes the alpha helix in the secondary structure. And further details if you give me, that between the NH and the CO, between the NH and the CO of the fourth amino acid ahead. I always say it's the first and the fifth. So 4 amino acids ahead. So NH and CO of the 4 amino acids ahead. So this is the first amino acid, this is the second, this is the third, this is the fourth, and this is the fifth. So hydrogen bonding between this and this. 1, 2, 3, 4, 5. So it's between the first and the fifth. Or you say 4 amino acids ahead. Then coming to the C part of the question, vaccination can be used to prevent the spread of cholera. The vaccine used contains part of the collagen molecule, and also contains dead cells of Vibrio cholera. So phagocytes play an important role in the immune response to a vaccine. Phagocytes contain many lysosomes. Describe the function of lysosomes in phagocytes. So they digest or they hydrolyze the pathogen. They contain hydrolytic enzymes and they fuse with the phagocytic vacuole. So basically, this is something which we study in the phagocytosis kahani in the story in the immunity chapter. Suggests the advantages of having a vaccine that includes dead cells of Vibrio cholera and part of the collagen molecule. So cells are dead. The cells are dead. So they cannot cause the disease. So they cannot cause the disease. So that is something which you should have picked up from the question, because they are dead, so they cannot cause the disease. And there is a part of the collagen molecule. Part of the collagen molecules, that part cannot be toxic. So part of the collagen molecule, so no toxic effect. So there's no toxic effect. Then another thing you could have said, then this is going to be recognized by the B lymphocytes. So this is going to trigger the clonal selection. So the B lymphocytes are going to realize, oh my God, this is a stranger. Now we need to be cloned. So trigger the clonal selection. Trigger the clonal selection of one type of B lymphocyte. Right? Then this will stimulate the production of antibodies. This will stimulate the production of antibodies, because then when the B lymphocytes are triggered off, then they're going to produce the plasma cells and the B cell, and the plasma cells and the memory B cells will be formed. And the plasma cells, P for plasma, P for produce antibodies. So stimulates the production of the lymphocyte, then stimulates the production of the different antibodies, and then it stimulates the production of more than one type of memory cell. More than one type of memory cell. So more than one type of memory cell. Why? Because you see, something is triggered by the dead cells and another by the collagen. So of course we have a very double response, and that is going to be a very, very good vaccine. Then coming to the last part, which is the D part, the World Health Organization states that most cases of cholera can be treated with oral rehydration therapy. Oral rehydration therapy is the ORS, which is just water, salts, and sugars, glucose in it. So people who have diarrhea and before they get dehydrated, we give them these ORS, and so they keep on taking that as a drink. Of course, you get it in flavored form as well. It's also called Pedialyte. So you keep on taking this, so your body immune system recognizes them and makes antibodies, and you don't have to take any other medicine. To reduce the risk of antibiotic resistance developing, antibiotics are only recommended for the most serious cases. So antibiotics are the ones which we take, like vibramycin, augmentin, amoxicillin, and all these. Now discuss the consequences of the development of antibiotic resistance in Vibrio cholera. Now, if they become resistant to the antibiotics, well, what problems are we going to have? We're going to have people dying of cholera, because we will be unable to kill the bacteria. So the first thing is that the Vibrio cholera is not killed by the antibiotic. So these people are going to be very, very sick. They're going to be very sick because the antibiotic is not going to work, because the Vibrio cholera has now become resistant to it. It's not killed by the antibiotic. So we'll be unable to treat people. People will keep on having the disease and they'll be suffering, unable to treat people with cholera. Unable to treat people with cholera using antibiotics. So there will be prolonged periods of illness. You're going to have long times you're going to be absent from school. Prolonged periods of illness, and the person, not school only, people will not go to work. Maybe people work in the hotel industry, maybe people work in some other industry, or so they work in a restaurant, so they will not be going to work. Prolonged periods of illness, increased risk of spread. There is increased risk that will spread to other people, because if we have continuously people suffering from a disease, so increased risk of spread. And then we have transfer of the antibiotic resistant allele to other species. So the transfer of antibiotic resistance to other species would of course be very, very even worse than this. So transfer of antibiotic resistance allele. to other species of bacteria. That is not a very good thing, because then we will have problems in other diseases as well. Just a few more points. Inability to treat other bacterial diseases, because you see, when we have this antibiotic resistance in cholera, and if this is transferred to other species which also cause disease, then we'll be unable to treat other bacterial diseases. Then increased death rate from bacterial diseases. Like, you remember, Quaid-e-Azam died of tuberculosis because there was no treatment then. Now nobody dies of tuberculosis because there's a lot of very effective treatments now. Then we will need to research and find out alternative antibiotics, and that's going to cost a lot of money. It's going to cost a lot of research, human labor. So this is all going to result in a lot of problems. Now this with this we come to an end of this video, and I will continue the rest of the questions in another video which will follow very soon. Thank you once again, and please do give me a feedback as to how I can help you more. Thank you.